Hello, hi. Welcome back to the part 2 of the Cellular Energetics lecture series. So, in the previous lecture, we were discussing about glycolysis. So, now this part I think it will be a short part because we are going to check what will be the condition of pyruvate or pyruvic acid. I have said that we can say it as pyruvate or pyruvic acid, like I said in the last lecture, acetic acid and acetate, because acetic acid is CH3COOH, but in the physiological pH inside the cell it normally exists as ions, so we call it as acetate. So pyruvate, after the formation, after the glycolysis process, a 6-carbon glucose has been converted by a series of 10 reaction or 10 steps forming pyruvate. Pyruvate is a 3-carbon molecule, so one 6-carbon glucose can give 2 molecules of pyruvate or pyruvic acid. So what is the fate of pyruvic acid or pyruvate, which means what will happen to pyruvate after the process? We know that it occurs in the cytosol, which is the fluid part of the cell. So it occurs in the cytosol of the cell, and the fate of the pyruvate is dependent upon the availability of oxygen. This should be underlined. The fate of pyruvic acid is dependent upon the availability of oxygen. If so, after discussing the three fates, the three major fates of pyruvic acid, this lecture will be completed. So our heading or the heading of the chapter today's lecture is fate of pyruvic acid. There are mainly 3 fates for pyruvic acid. The first fate, in the presence of oxygen. Actually, we can classify, I said that the fate of pyruvic acid is dependent upon the availability of oxygen. If oxygen is available, then it will be having a fate. If oxygen is not available, then it is having generally 2 fates in different organisms we will discuss. So first we will discuss the fate of pyruvic acid when the oxygen is available. I will draw the structure of pyruvic acid, if you remember the structure of pyruvic acid, because in the last lecture of glycolysis a series of 10 steps were involved. At last we got pyruvate, which is a 3-carbon molecule. So pyruvate is a 3-carbon molecule. I will draw the structure of pyruvate. 1, 2, 3. It is an acid, so CO minus H plus, because this condition is that it exists in as ions in physiological pH. This is the structure of pyruvate. Okay. If it is pyruvic acid, then we can write it as like this, but in normal physiological pH it exists as ion. So this is the structure of pyruvate or pyruvic acid. So what happens to pyruvic acid? Let us check. We can take it very easily. Pyruvic acid, I am first talking in the condition of oxygen. You should understand that we human beings or most of the eukaryotic organism are obligate in nature, which means obligate for the oxygenation, which means for the purpose of oxygenated respiration, which means that we need oxygen for our majority of the cellular purposes. But some organisms, like we have sometimes we can shift our oxygenated respiration to anaerobic respiration or unoxygenated condition. A good example is that when we are going for a good exercise, a very harsh exercise, in that condition the cell is lacking, our body is lacking plentiful oxygen. In that condition, for the purpose of respiration, for the purpose of production of energy, the cell needs the energy at a rapid speed. So what does a cell do? It undergo anaerobic glycolysis. That is the first fate of pyruvic acid, anaerobic glycolysis, which means after glycolysis continue, it will be having an anaerobic fate. So, but this is the case of some facultative organism, which means some organism can convert their metabolism in presence of oxygen and also in absence of oxygen. They can shift their oxygenation, their respiration process. But some organisms are always anaerobic in nature, like certain eukaryotes, small eukaryotes like yeast. They always follow anaerobic respiration condition. They can undergo anaerobic respiration condition. Whereas some organisms are always aerobic, some organisms are always anaerobic in condition. Like human beings, we can shift. We have dual mode of respiration. We have anaerobic respiration and aerobic respiration process. So in aerobic respiration, the fate of pyruvic acid is entering the next stage, which is the formation of acetyl coenzyme A. We will draw the structure. What is an acetyl coenzyme A? So that is the normal condition. Like glycolysis completed, pyruvate form acetyl coenzyme A. This acetyl coenzyme A is an intermediate for another, which means the second stage. I said there are mainly 3 stages: glycolysis, TCA cycle, ETC or the electron transport chain complex, and also the fourth one is the ATP synthesis in a pump. This is actually the usual process of respiration in organism, in aerobic organism or human beings. Normally it occurs. So the fate of pyruvic acid or pyruvate is dependent upon the presence of oxygen or absence of oxygen. If there is oxygen, normally if we are having oxygen in the cell, then pyruvate will form acetyl coenzyme A. We will draw the structure. After that, it will enter into TCA cycle or Krebs cycle or citric acid cycle. We will study why it is known as these names. It is known as citric acid cycle because when acetyl coenzyme A is an intermediate, oxaloacetate will form. So oxaloacetate and acetyl coenzyme A will be forming another molecule which is known as citric acid. This citric acid is an acid. Yes, obviously it is a carboxylic group and it is having 3 carboxylic groups within it. So it is known as tricarboxylic acid. We will study very deeply in an easy way for TCA cycle. Now our focus is the main step, conversion of pyruvate into acetyl coenzyme A. This only occur in the presence of oxygen. So we are going to write the structure and it occurs in the cytosol of the cell. You know, if oxygen is present, it will enter mitochondria and it will perform the next stage, which is formation of acetyl coenzyme A and TCA cycle and electron transport chain and ATP synthesis. But what happens in absence of oxygen? The pyruvate will be converted into lactic acid. We studied in the last lecture. We just discussed briefly. So we will first study the fates of pyruvic acid. Now I am talking about the aerobic condition. So in aerobic condition, In aerobic condition, pyruvate will be forming. What did I say? It is aerobic condition. It is a normal process. So pyruvate will be forming the next compound. This compound is required for the TCA cycle, an intermediate for TCA cycle. So we are talking about aerobic condition. Okay. So pyruvate will be converted into acetyl coenzyme A. Acetyl coenzyme A. Look, two different words are present. One is acetyl group and coenzyme A. What is acetyl group? We know that acetyl group is CH3CO. This is acetyl group. If we add OH, it will be acetic acid. If we add CHO, it is acetaldehyde. So this is acetyl group. So acetyl group and a coenzyme A. So what should we do here? Pyruvate should be converted into acetyl CoA, which means we have this structure. We should remove this. So carbon dioxide removed, COO. I hope it is clear for you. So carbon dioxide is released. So it will be having a decarboxylase enzyme. That is not required, but you just understand a decarboxylase enzyme means an enzyme, a protein molecule or structure, chemical structure which remove it. It is according to their affinity. So carbon dioxide is released. Now what should we add? A coenzyme A, right? First I will draw the structure of CO, SCOA, CH3. This is the structure of acetyl CoA. So a CoA should be added. So a CoA is added from a molecule SCOH. This is actually so CoA. This is actual structure of CoA. Actual structure of CoA, coenzyme A, which is a long carbon molecule. I will make you see the structure here. It will be coming. So a coenzyme A should be added to acetyl group. So this is known as acetyl CoA. It is done with the, so an acetyl CoA should enter cycle. So who make the mender or who promote the decarboxylation? This is all done by an enzyme. Okay, this is done by an enzyme which is known as PDH. PDH means pyruvate dehydrogenase enzyme. ACE means enzyme. It is done by an enzyme, pyruvate dehydrogenase. So this pyruvate dehydrogenase have the capacity to break carbon dioxide. It have the capacity to add this. It have the capacity to add acetyl CoA to group. What is this? S is actually the sulfur. Okay. So, and here what happens is that a H from here and this H, 2H is taken by an enzyme, taken by NAD is an enzyme, and it converted into NADH plus. So what literally happened is that when pyruvate was converted into acetyl coenzyme A, NADH is formed, carbon dioxide is released. It is done by the enzyme PDH. This is the only thing you need to remember. I hope it is clear. Why it is converted and what is the condition? The condition is oxygenated, plenty of oxygen available in the cell. So it can be converted into acetyl coenzyme A. Acetyl coenzyme A will enter TCA cycle. That is the coming part which we will be talking in the next lecture. Okay. Why acetyl coenzyme A? Because this acetyl coenzyme A has a many function. That is the reason we just, or the cell modify it into acetyl coenzyme A, because this acetyl coenzyme is actually an acetyl group donor. This can act as an acetyl group donor in the next cycle, which is the TCA cycle, which we will be learning very deeply. So if you just analyze it, a 3-carbon pyruvate is converted into 2-carbon acetyl coenzyme A with the help of PDH enzyme, with the help of, and carbon dioxide is released, NADH form. You know why these are formed? NADH I just discussed in the last structure, because last class, because these NADH are high electron carrier molecule, high energy electron carriers, which means they can produce ATP. NADH can produce ATP. There is also other function why this NADH is formed. So it will not promote glycolysis, but it can promote gluconeogenesis, which we will be discussing in the TCA cycle. But now in this lecture I will be only talking about the fates. This is one of the fate, formation of acetyl coenzyme. We have 2 more fates. First I will fill the ink. So this is the fate. One fate, if oxygen is available, and what happens to this, it will next now move into TCA cycle, where oxaloacetate is converted into citrate with the help of acetyl coenzyme. Only acetyl coenzyme can donate this acetyl group to citrate. So SCO get fixed with acetyl coenzyme. That is the reason. It is actually a cycle. It is known as cycle. It is converting, coming back. Why it is coming back, we will be dealing in the TCA cycle. So this is actually the first fate of pyruvic acid in presence of oxygen. This is to remember. The only thing to remember is that NADH2 is formed, 1 NAD is used, NADH2 is formed, carbon dioxide released. Okay, let me fill the ink. Okay, yes, I hope it is clear for you. Alright, so next we are going to deal about the second fate of the pyruvic acid, which is in the absence of oxygen. What happens if plenty of oxygen is not available in the cell and the cell need immediate energy? We know that ATP is the form of energy which the cell needs. NADH2 is also a form of energy, but it is not directly consumed. It is consumed by changing into ATP in the last 4th process, which is the ATP synthase process by a pump. It is actually a pump, F0F1 pump. Actually we have discussed in some of my video, in one of my lecture, where nerve impulse is being conducted. How nerve impulse is being generated and conducted, it is by certain pumps or channels in the neuronal cell. All cell have pumps, all cell have channels, but they are in particular cells. So this is the first fate. First fate means if oxygen is available, it can get converted into acetyl coenzyme A and it can get into TCA cycle. Then we are going to check about the second fate, where oxygen is not plentifully available. So what happens? So if you have analyzed, some of you may have doubt. Here no oxygen is being used, right? Here no oxygen is used, but they are undergoing aerobic respiration because oxygen is not used here. It is used in the last process, which oxygen is converted into water, which we will be discussing it in the coming part. If you are having confusion right now, leave it. I am just saying that because we are dealing about aerobic respiration, right? Aerobic oxidation process, but no oxygen is used here. So why? Oxygen is only used in the last process, not in the TCA cycle, even in the next process during the electron transporting, or known as oxidative phosphorylation. This is the process known as oxidative phosphorylation because oxygen is being used there. That is the reason. The third process is also known as oxidative phosphorylation. So this is the first fate of acetyl pyruvate. Next we can check the second fate. Okay, we are going to check the second fate of pyruvic acid, yani pyruvate. Okay, now what happens in anaerobic condition? Anaerobic condition where oxygen is not available. So what does the cell do? Cell then converts pyruvate into lactate or lactic acid. So let us draw the structure of lactic acid. COO minus or H plus is available here. COH, CH3. This is the structure of lactate or lactic acid. Okay, this is in the absence of oxygen, where oxygen, or not in the absence, it means oxygen is not plentifully available in the cell. We will discuss the application, what is the reason. Now just look to the reaction. In absence of oxygen, or where oxygen level is very low, the cell convert the pyruvate, which is formed by the process of glycolysis, into lactate or lactic acid. So if we analyze the structure, what we can find is that this O has been converted into H. So someone has donated H. It is donated with the help of an enzyme. NAD H plus get converted into NAD plus. So this is the condition, and it is with the help of an enzyme LDH, lactate dehydrogenase. It's actually, there is also presence of H here, so 2H has been given. It is with the help of an enzyme lactate dehydrogenase, so, or lactic acid. This anaerobic process occur in the muscle cells of eukaryotes, which means human beings. Muscle cells undergo mainly this anaerobic condition because muscle cells are a high energy demanding cells. They need every time ATP for their contraction and relaxation. Only when ATP is available they can contract or relax. So pyruvate is converted into lactate in the anaerobic condition. It is with the help of an enzyme lactate dehydrogenase. Yes, if you remove the hydrogen from group, it will be forming pyruvate. So lactate dehydrogenase. The enzyme name is given into any of the reaction. The name is given. So this is the condition. So why lactate is formed? Actually, it is not needed, but just understand why lactate is formed. The formed lactate is actually in the muscle cells. What they undergo is that pyruvate is actually formed. So why they are being converted into lactic acid? The reason is that now the cell is in anaerobic condition, which means the cell does not have the plenty oxygen. So pyruvate converted into lactate. Lactate now enter into the bloodstream, and lactate at the liver flows through the bloodstream, and in the liver it gets converted into pyruvate, and from pyruvate it will undergo gluconeogenesis, which is almost opposite to that of glucose, glycolysis. What is glycolysis? Formate of pyruvate from glucose. What is gluconeogenesis? Formation of glucose from non-carbohydrate precursor. So from pyruvate glucose is being formed. So now the glucose is available in the body. So glucose now enter into the blood and enter bloodstream and comes back to the muscle cells, so they can use energy very rapidly even though there is absence of oxygen. So this is the reason lactate is formed. High accumulation of lactate or lactic acid can leads to pain in muscles. This is the reason. Actually, this is the possible reason for the pain after a high exercise. Okay. So this is the second conversion in anaerobic mode. So in the muscle cell in anaerobic condition, NAD plus will accumulate. When NAD plus gets accumulated, here glycolysis can happen because glycolysis needs NAD plus. I think you understand, because we have studied NAD is needed for the conversion, right? So NAD plus accumulation is needed for the purpose of glycolysis. Okay. DHAP and glyceraldehyde 3-phosphate was formed from fructose 1,6-phosphate. Glyceraldehyde 3-phosphate should be converted into 1,6-bisphosphoglycerate. So this process need actually NAD plus. If cell does not have NAD plus, it will not undergo glycolysis. So in order to make the cell maintain their glycolysis process, pyruvate is converted into lactate, so that cell can undergo, muscle cells can undergo glycolysis. But what happens in liver, it is converted back lactate to pyruvate. So here NADH is formed, the reverse process, which means in liver cells amount of NADH is increased, which means for gluconeogenesis NADH is needed. So their gluconeogenesis occur, and in the muscle cells glycolysis occur. This is the reason. I think you are getting it. So this is the second fate, anaerobic respiration, lactic acid will be formed. Now the third fate, which is usually seen in most of the anaerobic organism like microorganism, where most of the time, which means their metabolism is anaerobic, where the organism where oxygen is not needed for their purpose of metabolism or respiration. So we are going to deal it. It is the third condition. Third condition means it is anaerobic, but it is seen in microorganism, anaerobic organism. Let us look what happens in anaerobic organism. I think I will write it with red pen or green. So what is the third fate of pyruvate? The third fate of pyruvate, which is seen in anaerobic organism, is the formation of acetaldehyde, and from acetaldehyde formation of ethanol. I mentioned what is acetaldehyde. CH3 CHO. This is actually acetaldehyde. CHO, CH3 CHO, acetaldehyde, aldehyde group. So an acid is being converted into aldehyde. Look, what is the difference here? We need an O, right? CO2 has been released. So the structure here left is CHO. So carbon dioxide is released first. So we need that probably decarboxylase enzyme should be present. So what is the enzyme name? Pyruvate decarboxylase. This is the name of the enzyme, pyruvate decarboxylase, which remove the carbon dioxide. We know that already H is present here, so no need of H there. Extra H. So carbon dioxide is released. This is known as acetaldehyde. So from acetaldehyde, what happens? Ethanol is formed. What is ethanol? CH3, CH2OH. CH3, CH2OH. So from acetaldehyde, ethanol is formed. What happens in ethanol? What should we need there? We need an extra H2, right? So this H2 is provided by NADH2 or NADH plus. Why we are giving this? Because in the structure H plus is loosely connected. That is the reason we are giving NADH, or you can say that NADH or NADH H plus. Okay, so an NADH is given, so the enzyme, so an ethanol is formed. The enzyme name is alcohol dehydrogenase, because a hydrogen is removed. So the name of an enzyme is alcohol dehydrogenase. This is the condition in microorganism. You know, this process is known as fermentation, because fermentation means anaerobic respiration or anaerobic metabolism of glucose. Yes, it is correct. What are the products here? Carbon dioxide and alcohol. So it is usually this process is usually used to make the beer or wine industry or beer industry, breweries. So yeast is used. Yeast is a microorganism, so it can undergo conversion, and some sugar is needed, right? Sugar means pyruvate should be formed, or glucose. Yeast should take carbohydrates or sugar. So in the process of wine industries or breweries, we keep yeast and also certain wheat, rice for the purpose of fermentation. Fermentation means anaerobic metabolism of glucose. So this is the process of anaerobic respiration, or the third fate of pyruvate. Look, pyruvate is being converted into alcohol and carbon dioxide is used, because in the cytosol of the yeast they release carbon dioxide, which is released outside of the cell, because we know that all gases are diffusible through the plasma membrane. Even though their plasma membrane are hydrophobic in nature, all hydrophobic gases are easily permeable. We have discussed in the pump, when we were talking about pump, how molecules cross the barrier, the wall of the cell, plasma membrane. So this is actually the third fate in microorganism, anaerobic respiration. So actually this is it was a small lecture which we were dealing about fate of pyruvic acid. I didn't connect it with the third lecture because it will be a lengthy lecture. So what were the fate? First fate was the formation of acetyl coenzyme from pyruvate. This is needed. This occur in the oxygenated condition. Then what happens in the second condition? Both two conditions are anaerobic. Anaerobic in the muscle. What happens? Pyruvate to lactate. Why lactate is formed? I said that because it can, in the liver, it can produce gluconeogenesis, and from that gluconeogenesis, glucose can be formed. This glucose can now enter muscle cells and undergo glycolysis. Glucose to pyruvate. So why they are undergoing glycolysis? Because ATP is released. That ATP is needed for the cellular machinery, enzymatic machineries. So that is the condition. Now the third fate is the formation of ethanol from pyruvate by a reaction, two-step reaction. Here carbon dioxide and ethanol, alcohol is released, which are used in the brewery, wine industries, alcohol industries. It is known as fermentation. So actually the anaerobic respiration is known as fermentation, generally if we are talking about, because it was made, it is the basic principle in wine industries. I hope this is clear for you. And why acetyl coenzyme is formed, even though there is no oxygen used in that step. But if acetyl coenzyme is formed, it can't come back to pyruvate. So What does the cell do in absence of oxygen? They will stop at pyruvate, because if oxygen is not available and if they convert it into acetyl coenzyme A, there is no use of acetyl coenzyme A, because it can't undergo into the next coming step, because the next coming step should need oxygen. So what does the cell do is that cell in absence of oxygen stop at pyruvate and look for the demand. If oxygen is not plentifully available, it will convert it into lactic acid in case of humans. In muscle cells where oxygen is not available, after a long run or during a long run, high energy is needed within a short time. So cells undergo anaerobic respiration rather than aerobic respiration, because even though oxygen is not available, it is not plentifully available. So this is the fate of pyruvic acid, the three main fates of pyruvic acid. So I hope this part is clear for you. We will see in the next part, which is the third part, we will be dealing with the TCA cycle, tricarboxylic acid cycle, a molecule which is having tricarboxylic group, acid group. I think you are understanding it. So we will be seeing in the next part. I hope all the lecture is clear for you. We will see. Okay, bye.